జాయిన్ అవుతుంది అందువల్ల స్క్రీన్ షేర్ చేయాలి కదా.
Ravi Kumar sir, over to you, sir. You can start.
Ramakrishna sir, shall I start the session?
JSA and intramural core is fine.
Ramakrishna sir, can you hear me?
Sorry, light last sir.
So, yeah. So once again, good morning all the delegates and speaker. So I am Dr. Ravi Kumar, Associate Professor, Sri Vishnu College of Pharmacy. I welcoming you all back again to the fourth consecutive day of this ACT sponsored STTP on advances in pharmaceutical formulations and regulatory pathways globally for small molecules, medical devices, and biologics, conducting by Sri Vishnu College of Pharmacy, Bhimavaram. So now I take the privilege to introduce the second speaker of the session today, Dr. Ravi Kiran, STTP. So he's basically PhD in biochemistry and molecular biology from Wayne State University, Detroit, USA, and MS in molecular biotechnology from Wayne State University, Detroit, USA, and MSc in biotechnology from Andhra University, Visakhapatnam, and BSc in botany, zoology, and chemistry from Andhra University, Visakhapatnam. So some of his professional accomplishments are Startup Laboratory in Rajahmundry, India, which is the Center for Advanced Applied Biological Sciences and Entrepreneurship. So startup company in Toronto, Canada, the EDD Institute of Discovery and Education. So he is a scientist FDBT India, structural biology team leader for breast cancer research at the Institute for Stem Cell Science and Regenerative Medicine, Bengaluru, and 20 years of biomedical research experience in USA, Canada, and India. He published more than 20 peer-reviewed scientific articles in international journals. So mentored more than 50 students, including doctoral students. Initiated more than 12 international collaborations, USA, Canada, UK, Japan, India, and successfully completed most of them, resulting in publications. So he is recipient of prestigious US federal government sponsored fellowship in the Intramural Cancer Research Training, Washington, DC, USA. So he deposited more than 30 X-ray crystal structures of proteins and protein drug complexes. So this is a brief profile of today's speaker, Dr. Ravi Kiran. So we are very thankful to you, sir. Now session is over to you. You please take over the session.
Sorry, I had a minor network issue.
Thank you, sir. Yeah, you can take over the session. I introduced your profile to the participants. Please take over.
Okay, thank you very much. Thanks for the kind introduction. So my name is Ravi Kiran. So I am going to share my screen now.
Okay, sure.
I hope you guys can see my screen.
Yes, sir. Yes, we can see the screen, sir.
Okay, great. So first of all, thanks for having me as a part of this meeting, and I feel privileged to talk about what we are doing in our lab in Rajamahendravaram and also in Visakhapatnam. So I'm the founder of TCABSE, the Center for Advanced Applied Biological Sciences and Entrepreneurship. So the goal was to make sure that we bring in the cutting edge research to India. So I was trained basically in the United States of America. And my goal is to bring those standards, the global standards, to India. So we are primarily a laboratory-based training lab. So we are interested in training mostly the undergraduates and the postgraduates, because most of the time that's where the training starts. I hope you guys don't mind if I give you like a brief introduction. Our main focus is to train the students as scientists and also scientific entrepreneurs. Many of you already know, people are getting trained and they go into the job market to, compete with other people for jobs. So one of our mottos is to create scientists, but also to create jobs. So we encourage all these students that come to us for training, not only to get trained in the science, but also to think about starting companies. So we tell them that as long as the novelty exists in a given idea, there's always a startup. So we started off in 2019 in Rajamahendravaram as a headquarters. And so we are a nonprofit lab. So I'm doing this out of my passion to research. So, and also, like I said, the goal is to bring the global standards back to India. And so we indulge primarily the undergraduate students and the postgraduate students in cutting edge research. We not only train them from the basics, but we also indulge them in critical thinking and also try to encourage them and also support them, when it comes to coming up with new ideas, designing new projects, and working on these projects. And also we include things like training in grant writing and how to get the funding, how to talk to the private investors to get some investments into your company. So we do a lot of things, and the idea is to train students as better future scientists, and also to train scientific entrepreneurs so that they can, in return, create more jobs so that we don't have too many people fighting for jobs in the job market. So the headquarters of TCABSE is in Rajamahendravaram, and we are working on different kinds of projects, anywhere from basic oncology to infectious diseases, metabolic disorders, stem cell biology, multi-omics, synthetic biology. So synthetic biology would be my focus of today's talk. You'll find out pretty soon. And we recently started a branch in Visakhapatnam. So the idea is to provide the students with the opportunities and the proper guidance, and also the appropriate equipment and the laboratory procedures so that they can be trained well, and also give them a chance to think about how they can take an idea to a startup company. And then we encourage the students to do this on a daily basis. So we have successfully trained more than 70 students so far, from both the Rajahmundry labs and also the Visakhapatnam lab. And I'm happy to inform or say that we're venturing out into the clinical research in the near future. So far, we have established our laboratories on basic science research, but then the future will be all about the clinical research. So as you can see, these are all my trainees, and most of them are indulged in real-time projects. Okay, so enough about my lab. So let's get back into the formulations. So I'm pretty sure most of you already know the details that I've put on this slide, but I just wanted to make sure that everybody's on the same page. So we have in classical pharmacology, we have the small molecules or ligands or drugs that bind to the receptors, and they create the pharmacological effect. And also there are the dose response curves where we look at CC50, EC50, and also define the therapeutic window. All of these will, the major challenge that we usually face in most of these things is the bioavailability and the ADME tox. I'm sure everybody knows about these things, so I don't need to explain the details, but I just wanted to, start off from the basic. And the pharmaceutical formulations. We basically do this to protect the compounds, or try to do the controlled release. So again, it goes back to the oral bioavailability, because if the actual ingredient is not bioavailable, or if the bioavailability is not that great, so the formulation may or may not help. And also, when it comes to cancer, oncology, anti-cancer drugs, this is where we have the biggest issue, because typically in the infectious diseases, the drugs that we administer, they go directly and target the pathogen, the bacteria or virus or fungus, which is the target molecule is pretty much non-existent in human. But when it comes to cancer, unfortunately, we're targeting our own proteins. So this is where selectivity becomes an issue. And so systemic toxicity is primarily a big concern when it comes to the anti-cancer drugs. And I just wanted to just to reiterate that we could come up with the best formulations or the best delivery systems, but the systemic toxicity is still an issue as of today, and it needs to be addressed. So, for example, let's consider cisplatin. It's a great drug. It causes apoptosis in the cancer cells, and it works great. But the biggest problem that we typically deal with in the cisplatin regimen is it can also target the healthy cells. So obviously, this is what I mean by systemic toxicity. And so, I mean, as you can see in the diagram, the cisplatin, it gets directly into the DNA double strand and confuses the DNA polymerase, and there will be base mismatch. And this base mismatch will end up with mutations, and these mutations will ultimately lead to the apoptosis. So when it comes to the apoptosis of cancer cells, that is great. But if it's the apoptosis of healthy cells, that's where we have a problem. So this is where the systemic toxicity comes into play. And so the idea or the point I'm trying to make on this slide is that we need an alternate or a better formulation or better delivery systems so that we can avoid, if possible, or at least minimize the systemic toxicity of these drugs, primarily in the cancer or the anti-cancer drugs. I mean, some of the drugs are specifically targeting specific enzymes, but drugs like cisplatin, it is targeting DNA, which means it could target any DNA in any of the cells, and that is highly toxic. Okay. Then the big question comes is, how to solve this severe systemic toxicity. So we have multiple anti-cancer drugs. They're all great drugs, and they've been proven both in basic research, clinical research. But the problem is most of them fail when it comes to the systemic cytotoxicity. And this is where a lot of patients are also losing their battle against cancer. I mean, some of them recover, but some of them end up with a relapse. Some of them lose the battle. So I think systemic cytotoxicity is creating big problems, and we definitely need to address this question. Going back to a cisplatin example, as I'm showing this on this slide, you can see that one single cisplatin drug can create so many different systemic cytotoxicities. So this is a serious issue that needs to be addressed quickly, especially in the cancer biology. So this is where my lab, so we have our intramural research. So we are working on synthetic biology. So here I'm showing you a picture that I took it from Oxford University Press. So this is a electron micrograph of E. coli Nissl 1917. So this is a strain, a special strain. So E. coli Nissl, it's a commensal, and it's commonly seen in the human gut. So this is an old friend to the humans, so it is safe. And so what we're trying to do in Visakhapatnam is we're trying to take advantage of the E. coli Nissl strain 1917 to bypass the systemic cytotoxicity. So that we're using synthetic biology route basically to achieve two things here. Number one, to reduce the systemic cytotoxicity, and also the second one is to have a precise and controlled delivery and release of the drug at the site of action, which is what is missing when it comes to the regular classical pharmacology, because an oral drug has to go through the metabolism, and everybody knows the SIPs in the liver. And then you get all these metabolites that would end up with drug-drug interactions in liver, and also they cause kidney toxicity. So if we can come up with a system, preferably with the synthetic biology, we can bypass a lot of these issues, and also primarily we can decrease the systemic cytotoxicity of the anti-cancer drugs. My lab is heavily indulged into the synthetic biology. And so what we're doing is we're starting with this E. coli Nissl 1917. So this is our parent strain. And so we are further genetically engineering and we're further modifying this strain. So before I go any further, let me just give you an example. For example, this is a paper that was taken, that was published by Chen et al. So as you can see, people have, in the past, people have actually used the E. coli Nissl 1917 as a probiotic. And as you can clearly see in this diagram, so they created a matrix on a silicon chip. And to this matrix, they have the E. coli Nissl biofilm. And because of this biofilm, the E. coli Nissl would actually avoid pathogenic bacteria or any other kinds of pathogens to, enter the guts. I mean, this will become more obvious in the next couple of slides where I'll be discussing more details. But the point that I wanted to make on this slide is that E. coli Nissl is completely safe and it is commonly used as an probiotic. And as you can see, they also have a brand name, Butaflor. I think this is from Australia. Okay, so in this slide, I'm giving you an example of how people have successfully genetically engineered the E. coli Nissl. So, for example, on the left side of the slide, you can see there is an inflammation in the large intestine. And so one of the approaches is to create a patch by using some proteins. So if you create a patch so that the inflammatory cells can be controlled, so that the inflammation can be controlled. So in this case, as you can see in the diagram that says D, panel D, there are two sections that are showing minus patch and plus patch. So without the patch, you can see all the blood cells are getting into the lumen. And that's probably causing a problem for the inflammation. It's actually making the situation even worse. But when these guys have actually genetically engineered the E. coli Nissl that can create the patch, and the patch is shown in like a strands, the yellow and green strands. So the patch actually covers up the site of damage so that there is a decrease in the inflammation. And further going into the details, the molecular details of this experiment that they have published, they've taken the E. coli Nissl and they've genetically engineered. They've included a plasmid that is controlled under a inducible promoter. And so this bacterial cell is genetically engineered so that it constantly expresses the therapeutic curly hybrids. So basically, these are proteins that are repeatedly generated in the gut by this genetically engineered bacteria. So by doing this, these therapeutic curly hybrids, they're creating a patch so that the patch would actually control the inflammation or the infiltration of the inflammatory responses so that the inflammation is gradually controlled. So this is a fantastic example of what people have already done. And as you can see, this paper is published in 2019, which is pretty recent. So we got really inspired by this system. And then we asked questions like, why not we use something like this? And why not we ask the bacteria to synthesize the drug at the site of action. So for example, let's say, lung cancer or kidney cancer or any type of cancer. So we genetically engineer the bacteria so that we precisely deliver the bacteria to the site of action or the tumor microenvironment, so that when the bacteria goes there, it actually senses the tumor microenvironment, and then it gets triggered to synthesize the drug and of course release the drug. So this is what we are heavily involved in as of today. We have chosen cancer as our first choice because anti-cancer drugs are extremely toxic when it comes to the systemic toxicity. So we have taken the E. coli Nissl and we are in the process of genetically engineering them so that they can go to the tumor microenvironment and they can sense the chemicals in the tumor microenvironment. And the sensing of those chemicals will trigger the promoter that is built in the E. coli missile, so that the E. coli missile will express the required proteins. And so it would be a cascade of enzymes, so that these enzymes would actually synthesize the required drug molecule at the tumor microenvironment, or, in other words, at the site of action. So by doing this, we're bypassing the whole systemic toxicity, because the drug is not released into the blood. It is only synthesized and released at the site of action. So we don't need any special formulations, or we don't have to worry about the bioavailability. Of course, however, we have to worry about the drug-drug interactions, because even if we precisely deliver the drug at the site of action, we may still have some metabolic issues, because all the drug molecules eventually go into the liver, get metabolized. But by using this approach, we believe that we're significantly decreasing the systemic cytotoxicity, which has been the major issue in cancer biology. In addition to the synthesis, another aspect that we're highly interested in is the controlled release of the drug. So once the genetically engineered E. coli cell synthesizes the drug molecule at the site of action, then the drug must be released by the E. coli cell in a controlled fashion. So we have identified genetic circuits so that we can control the expression of channels that would pierce through the E. coli cell wall, so that the drug can be released. So that expression and upregulation of those channels are tightly controlled by using genetic circuits. In this way, we are bypassing the systemic cytotoxicity. And of course, we're open to collaboration. So if anybody thinks that this is a cool idea, please talk to me or contact me. That would be fantastic. Okay, this is another paper that I wanted to give you, because this paper, whatever they have published in this paper, is more close and more relevant to what we are doing at TCABS-E in my lab. So like I explained earlier, so we designed the E. coli Nissl strain. So this is the E. coli Nissl strain. And primarily, the first thing that happens is there is an external detectable signal. So this could be some cytokines, or this could be some chemicals coming out within the tumor microenvironment. So these chemical signals are detected by the synthetic cell, and then those will induce the promoter. So the genetic circuits could be either in the chromosomal DNA, or they could be on a plasmid DNA. So that's an additional benefit, the flexibility that we have in the E. coli system being a prokaryote. So of course, when the signal binds to the promoter, and we have the coding sequence that is getting expressed, and we have the proteins that could be targetedly released at the site of action. So this diagram here basically explains how one can do it for proteins, anti-cancer proteins, such as growth hormones or whatever. But then also the interesting part is you can do the same thing for RNA delivery, or product delivery, or cytotoxic agents near the tumor microenvironment. So this is where we're particularly interested in. This is what we're actually doing. So you would have a tumor here. You can see the blood vessel, and the cells are growing. And if we can send these synthetic cells right to the site of the tumor, or the tumor microenvironment, these cells will specifically detect the external stimuli. And then accordingly, the genetic circuits will be turned on, and then the required drug is synthesized and released in a controlled fashion. So this is what we're working on as of right now. And without going into too many details, in the interest of time, I just wanted to give you just an overview of how all of this happens and how we design these things. So as it is very clear, I found this diagram from the publication from Higashokini from Current Opinion in Biotechnology. So we have the inputs, the extracellular environment, or any of the chemicals, ligands. And so we design sensors. So sensors are basically the promoter DNA of the genes that we build into the E. coli, the synthetic cell. And these sensors would link to the effectors. So the effectors are basically the proteins that could be directly released in the tumor microenvironment, or these proteins will in turn synthesize the drug within the cell. So this could be a cascade of enzymes that would catalyze different steps of drug synthesis within the E. coli cell. And then, so the signaling networks, this is where all the complexity is, because there's a lot of genetic circuit design involved. As I mentioned here, and/or. So this kind of takes us back to our basic electronics classes when you're in the high school, probably in the 10th standard when you're studying the physics. So the synthetic gene circuits also work in the same way. So for example, let's say the E. coli Nissl is in the tumor microenvironment. We design the genetic circuit in such a way that at least one or two of the external signals would trigger the genetic circuit. So it has to be external signal 1 and 2, otherwise nothing will happen. So if the external signal 1 and 2 triggers the promoter, then you'll have one protein that gets expressed. And when that protein gets expressed, then we design another downstream genetic circuit that gets turned on based on what the activity of this protein is that is getting expressed from the first circuit. So in this way, we have to generate a complicated network. So this network basically contains all the enzymes that you need to catalyze the synthesis of the drug within the E. coli cell. So once we finish, then we have the outputs. The outputs are basically drug molecules. So once the E. coli cell synthesizes a drug molecule, then it is successfully released through a channel. So we have engineered a channel, and this channel lets the drugs get secreted in the tumor microenvironment. Of course, we have a collaboration, so we're working with them on this. And so we can control the amount of drug released at the site of action after the synthesis, again with an and/or gates. So I mean, I'm saying gates, but they're like circuits. So the genetic circuits. So finally, you can secrete the drug in nanomolar or micromolar. So you can adjust this by adjusting the level of the channel expression. Again, that is another genetic circuit. So you can basically determine the dose of the drug that is released at the tumor microenvironment post-synthesis, and it is all tightly controlled. And the beauty of this is if the E. coli Nissl, or if the synthetic cell does not find any external signal, then nothing will happen, and it stays as a probiotic, or it's just a commensal, stays in the human body. And because it is a commensal, it stays under the radar of the immune system, so it doesn't trigger the immune system, unless there is in certain cases, it may attract the immune system, which is also works for the benefit of acting against the tumor microenvironment, because we are basically triggering an immune response against the tumor cells. But most of the time, because the E. coli Nissl is a commensal and it is under the radar, so nothing will happen. So if there is no signal, external signal coming into the cell or triggering the cells, the genetic circuits remain silent. Nothing gets expressed, and the cell is just going to swim around and nothing will happen. So this is completely safe. And so this is how we are planning to bypass the systemic cytotoxicity, primarily when it comes to the anti-cancer drugs, which has been the biggest challenge, both from a formulation point of view and a drug delivery point of view. Okay, so I guess this is all I have. I mean, we do have some data that I cannot share at this time, but in future, definitely, if there is another opportunity that shows up, I'd be more than happy to share all the data that we have, and then we'll be publishing all our data, so it'll be out there for you to go through. I'm going to just summarize what I have discussed. So synthetic cells, so we're basically building the genome of this E. coli cell, the E. coli missile, in a way that it works for us, and we precisely control the genetic circuits. And so the way how these are designed is so that you have a sensing genetic circuit that would respond to the external stimulus. Such as it could be any chemicals that are released in the tumor microenvironment, or any of the cytokines or interleukins and so forth, or receptors. And then the second step is there is a synthesis of the drug. So the second genetic circuit gets triggered. And once the drug is synthesized, then it gets released. So that's the third genetic circuit. So it's all combined in a very tightly controlled manner, so that the cell would function completely only if needed. Otherwise, nothing will happen, and it just stays as a commensal. As I mentioned earlier, the bioavailability and systemic toxicity of these anti-cancer drugs has been a major issue, but by using the synthetic biology route, I am pretty confident that we can bring those into acceptable levels. And also, we don't really need to worry about the formulation, because the active ingredient is synthesized at the site of action and released in a dose dependent manner. So when you have the active ingredient directly going through, we don't have to worry about getting the capsule or the tablet going through the stomach in the acidic environment and all those things, or the stability of the drug, because the drug that's synthesized may or may not be released immediately, because we can control that too. Like, for example, we can control a particular drug getting released for maybe today, and then we can control it and getting released probably after 2 days or something like that. So there's this is all about a series of genetic engineering and genetic circuits. So you can create as tight regulations as you want, and that's why this is a pretty exciting option. And the synthetic biology platform, it could be extended for other purposes in pharmaceutical industries, and not just drugs. You can also use these for proteins, and which brings me to our future goals. So one of the two future goals that we have in mind is, besides the drugs, we also want to extend this to prodrugs. We want to extend this to biologics, and also the most exciting thing that we wanted to do is to use the synthetic biology for RNA delivery, because microRNAs have been excellent therapeutics so far, but the biggest issue with the miRNAs is the delivery and the formulations. But with this system, we don't have to worry about that, because the microRNAs will be synthesized by the synthetic cell at the site of action through the tightly regulated genetic circuits, so that we don't have to worry about the systemic toxicity of these microRNAs. I hope I made it clear, but I'd be more than happy to take any questions, and thank you. And here's my contact details if anyone's interested in collaborations or any further questions. And I would like to encourage you to visit us on the web. You can find more details on the web. Thank you.
Thank you, sir. Indeed, it is a very good presentation, concise presentation. I see you have given very elaborate areas what you're working on very precisely. So before I request questions from the Zoom audience, so can I ask you doubts or clarifications, sir? So can I read? Yeah. So it seems like your area of, your lab established currently at Rajamahendravaram is more or like in synthetic biology and biotechnology oriented works are going on. So what is the scope of pure formulation and pre-formulation scientists from the pharmaceutical R&Ds and academia to collaborate with you, sir? Can you briefly?
Yes. So, like I said, primarily in the context of anti-cancer drugs. So any pharmaceutical scientist or any industry scientist, if you have a new compound and if you are facing the bioavailability admin tox issues with it, I would suggest you try the synthetic biology route because that may solve all your issues. So it could be, small molecules, it could be prodrugs, or it could be anything, nutraceuticals or anything. So I guess, this is an open platform that anybody who's in drug discovery field can exploit and take advantage of.
So about the areas of work, you said about you are emphasizing more on anti-cancer drugs, oncology area. So can anybody approach you for antimicrobial activities, kind of things, to go for to have any proof concept studies, something like that?
Yes, absolutely. So we are working on HIV. We're working on COVID-19. So I guess, just to give you an idea of how interdisciplinary we work in the lab. So at the moment, we are using the artificial intelligence-based, machine learning-based predictions to design a COVID-19 vaccine for the second generation, just in case if there is a mutant form of the SARS coronavirus 2 in the future. So I mean, the typical classical way of designing vaccine is you take a protein and you just inject into the mouse or in humans and generate the immune response. But what if there is a mutation? What if there is a change in the structure of the antigen? So this is where we are using the artificial intelligence to make predictions of how many different types of shapes then a given antigen can take, whether it is coming from COVID-19 or bacteria or TB or anything. So definitely, yes, antimicrobial activity is one of the things that we do very commonly.
Yeah, it's a great contribution, sir, from your side to the colleges and the scientists, principal scientists, especially working at the college level, because it would be always difficult to collaborate with the companies, lab like entrepreneurial labs like TCABS. It's very helpful for us.
Yeah.
So similar kind of query is, can you have any helping hands for the implementation of grants? So actually nowadays central government is encouraging a lot of grants for the academia people. So some of us are working on the writing proposals, and some of us also got the grants. So in those cases, do you have any specific plan to help us help these kind of people in academia?
Absolutely, why
Cross.
not? Absolutely. I mean, I have already mentioned that we are a nonprofit organization. So we're definitely interested in collaborations and also writing grant applications. We can help you. So I guess, the one way of doing this is, we can initiate a collaboration between your lab and my lab, and then we can come up with a proposal that works for the best of the interests on both sides. And absolutely, we can. And so at TCABSE, we not only apply to the Indian government, we also write grants to Washington, D.C. So we are also applying for grants from Washington, D.C., so especially for the undergraduate students and postgraduate students. And we want to showcase them that, these are the future scientists. So the answer to your question is yes, absolutely.
So it is a very useful question, sir, for many delegates and audience, because nowadays collaboration projects are encouraging by the central government for
Right.
many project grants.
Right.
So yeah. So there's one question from audience, that is Dr. Nalanda is asking, is the allosteric gene regulatory element can be considered are useful for the cancer research?
Yes and no, because it depends on the type of the cancer. It depends on the type of the gene. It depends on if you're talking about an oncogene versus tumor suppressor gene. The answer is yes, definitely you can do that. But if you are proposing the E. coli Nissl is a prokaryote, so it's easy to manipulate. But if you're thinking about a eukaryotic cell, such as yeast, yes, it still can be done, but it is much more complicated. I hope I answered your question.
Yes. So I
Very good
request
morning,
Dr.
Dr.
Sunita
Ravi.
madam, would you
I
like
have
to
few
ask?
questions for you. Can I continue, Dr.
Yeah,
Ravi?
please. Yeah, please, ma'am. Please go ahead.
Yeah, it's a very nice presentation. I'm
Thank you.
Dr. Sunita, presently working at Geetham School of Pharmacy, Hyderabad campus. So I have a few questions for you, Dr. Ravi. The first thing is that is the probiotics same as it of the dietary supplements, or is there any difference?
So there are several probiotics, and we are specifically interested in only the E. coli Nissl because it has been worked on by several groups internationally over the globe, across the globe. So we feel confident and safe because there is a lot of data availability on this specific strain, sorry.
The question is the probiotic is a part of the dietary supplement, or is it comes into the separate category?
So probiotics are more to create a healthy microbiome. So I guess the best way to answer this question is, as you already know, the human gut contains a lot of microbiota. And so it's all about having the right bacteria. So if you have the right combination of bacteria, then you're considered healthy. And if you have the wrong combination of bacteria, then it could probably lead to complications. So in order to maintain the healthy gut, you have to have healthy bacteria. And so that's where the probiotics come into play. So probiotics are primarily, E. coli or bacterial cells. So these are not dietary supplements. So dietary supplements are more like molecules. They could be vitamins or, minerals or something like that. But probiotics are primarily cells, E. coli cells. Or modified, bacterial cells.
Okay. How about the use of the probiotic? If it is used for long terms, are there any side effects? Are they safe?
So by definition, probiotic is, it's improving your health. So as long as the manufacturer, whoever is supplying, has a good strain of bacteria, we should be fine. So using probiotics is always a good idea. But when it comes to the, let me just back off a second. Let me clarify. So in the Western world, things are different. But when it comes to India, the buttermilk is one of the most common ingredient in the food. It doesn't matter which part of India it is. Throughout the India, we drink a lot of buttermilk, which contains healthy bacteria. So do we still need to take probiotics? Sure, why not? But I would just suggest drink buttermilk every day. I hope I've clarified your question.
Yes, because when I have gone through the probiotics, it is reported that for a longer time there may be some side effects of the probiotics. So.
Yes, it's all about the balance, right? You definitely need the bacteria in the gut, but you don't want too many bacteria. So that could lead to something else. I guess you just answered your question. So yes, a long time usage would probably populate too many bacteria in the gut, and that might probably create, the imbalance and the physiology.
Okay. So how you are going to do the safety and efficacy of your probiotics? Is there any specific guidelines do you have, or how you are going to do the safety, efficacy, as well as the stability?
So safety and stability are already done because they have been used as probiotics. So I'm not worried about that. And the engineering part of it, the design part of it, is something that we are currently not able to release for some reasons. But like I said, in the near future, we'll be publishing all the details.
Right. Okay. Thank you very much.
You're welcome.
Wish you all the best.
Thanks.
Thank you, Dr. Sunita. So, Dr. P.V. Swami, sir, would you like to ask a question? It seems like you want to ask. Okay. So, I think many questions and queries are already answered, sir, which seems like similar kind of questions are coming in YouTube chat also, but you already answered those questions when I asked at the beginning. So now So can you hear me, Ramakrishna sir?
Sir, there is another question in Swami sir, from Swami sir. Sir, please read out that question, sir. Zoom chat you can see. Ravi Kiran sir.
I don't see.
Yeah, the question is from Dr. P.V. Swami sir. Are probiotics helpful in improving immunity in the present situation?
I'm sorry, can you repeat the question, please?
Yeah, are probiotics helpful in improving immunity in the present situation?
The simplest answer is no. The long-term answer is yes, because taking the probiotics will restore the intestinal health. And of course, intestine is where you have the microvilli, where all the food is taken in by the blood vessels and circulated. Whether it is your immune system or cardiac system or neuronal system, any system, in order to be healthy, you have to have a healthy gut, because everything is ultimately related to the gut. So I guess my answer to the question is there is no magic bullet that you can just take today and then become immune to anything and everything tomorrow. It's a gradual thing that you have to build over time. I hope I answered the question.
So Ramakrishna sir, our questions are completed.
Yes, sir. From YouTube, I posted the questions already. I hope you asked them.
From Zoom audience also.
Yes.
So sorry for network interruption. Once again, I request Nagarjun sir to continue the vote of thanks, formal vote of thanks.
Thank you, Ravi Kumar sir. Once again, it's my privilege to propose vote of thanks for this ICT-sponsored one-week online short-term training program. On behalf of Sri Vishnu College of Pharmacy and Management, I extend my hearty gratitude to today's speaker, Dr. Ravi Kumar Y, founder, principal scientist, Center of Advanced Applied Biological Sciences and Entrepreneurship, Rajahmundry, for his talk on the following topic: future pharmaceutical formulations with biological risk. Sir, you have given a valuable information on this topic, and our viewers has gained knowledge on synthetic gene circuits, synthetic cells containing gene circuits, and also the bioavailability factors in your presentation. I express my scientific thanks to our director, sir, and also my sincere thanks to our director, sir, Dr. Kumar V.S. Nimmani Garu, for his efforts, for his proposal to bring such eminent personalities and also introducing for us by your speaking, by your presentations also. Our sincere efforts will, sir, your sincere efforts will gain knowledge on the scientific talks like you. And I also thanks K.S. Natarajan Garu for giving this, for also supporting us to conduct these scientific sessions in a smooth manner. I extend my thanks to my coordinators who are working off the screen, especially Mr. V. Ramakrishna Garu and Dr. Bhakiraj Garu. Last but not least, I take opportunity to thank our chairman, sir, Dr. K.V. Vishnuraj Garu, and our vice chairman, sir, Dr. Ravi Chandan Raj Gopal Garu, and secretary for Sri Vishnu Educational Societies, Sri K. Aditya Visham Garu, for providing this opportunity. At this juncture, I also thank all the participants for who have extended cooperation and also acquired significant knowledge through this session. Thank you, sir. Over to you.
Thank you, Nagarjun sir. So thank you, Dr. Ravi Kiran, once again for your kind acceptance of our invitation and given very concise presentation.
Thanks for the invite.
Dr. Ravi Kiran, sir.
Yeah, so I guess there was a network
Yes,
issue.
there is a network issue with Ravi Kumar. I hope. Thank you very much, sir, for being with us today.
Yeah, thanks for giving me the opportunity.
Thank you, sir.
Yeah, bye.
Yeah, I'll be signing off for you today. And tomorrow, please do join us for talks from eminent speakers such as Dr. Tanga Mariyappan, Ms. Fareen Ahmad, and Dr. Geeta in the afternoon session. Please do join us at 9:30 a.m. tomorrow. Thank you all.